Yeah, thanks, Dirk, for the introduction, and I would like to thank the organizers for inviting me to this interesting meeting. What I actually would like to do is to present you a new model where just by bioinformatic analysis we aim to identify target antigenes that potentially could be used as kind of a therapeutic, but also potentially as a preventive vaccine for certain subtypes of colorectal cancer. And the idea behind that project was that each cancer has to be occur through a pathway that induces genomic instability. So this is the major pathway how all cancers occur. There are two major molecular pathways. One is chromosomal instability, where in those tumors there are more random events, chromosome breakages, mutations that occur through various types of DNA repair defects and other things that lead to a fairly unpredictable level of mutations. And there's a second group of cancers that occur through the mutator phenotype, or the microsatellite unstable cancers that are actually all due to a complete lack of DNA mismatch repair activity in the emerging cancer cells. And particularly the second mechanism is quite interesting because you know the mechanism how the DNA is changed. There is virtually no major chromosomal damage occurring in those tumors, and simply by analyzing the genome and predicting mutations that occur in certain sequence stretches here, one presumably would be able to predict mutations that can be immunogenic in that context. Now, it's not an irrelevant mechanism. It's not rare. About 15% of all colorectal cancers, and many of the other cancers too, arise through this DNA mismatch repair deficiency system. 53% of those cancers are actually belonging to the hereditary non-polyposis colorectal cancer syndrome, which is the most frequent inherited cancer syndrome at all. And there's about 12% of all colorectal cancers develop this DNA mismatch repair deficiency just by sporadic mutations or sporadic epigenetic events, usually in those cells where the DNA mismatch repair genes are silenced by epigenetic mechanisms. Now, these tumors are quite interesting in terms of tumor immunology because there's one clear hallmark for those cancers, and this is a very dense infiltration by lymphocytes. This is actually a pathogenetic hallmark used by pathologists. If in tumors such dense infiltrates are detected, there's usually a very high chance that those tumors are indeed belonging to the DNA mismatch repair deficient cancers. And the second major aspect is that these tumors who occur through this DNA mismatch repair deficiency pathway are usually displaying a substantially better prognosis compared to the microsatellite stable cancers that occur through the chromosomal instability pathways. So obviously, something is going on in these tumors that attracts the attention of the immune system, leading to these type of immune infiltrates. And on the other hand, this at the same time is linked to a substantially better prognosis of those cancers. Now, the mechanism how these cancers occur is simply complete lack of DNA mismatch repair activity, and this is usually achieved by the inactivation of one of the components of a complex, the DNA mismatch repair complex, that safeguards the sequence identity after each round of DNA replication once these cells are dividing. So if one of those proteins is missing, the complex can't work any longer, and the consequence is that the cells survive, but they accumulate fairly rapidly mutations in their genomes, and these mutations are usually, by thermodynamic reasons, linked to repetitive DNA sequences, and these repetitive DNA sequences are usually referred to as microsatellites, mononucleotide sequences in particular, so that there is an accumulation in repetitive DNA sequences fairly rapidly accumulating in these cancers, and this led to the term microsatellite instability because these microsatellite sequences are the primary target of those mutations then. Now, microsatellite sequences can be found in the genome anywhere. It's in the intergenic sequences, it's in the promoter or intron sequences, but most importantly, it also are frequently found in coding exon sequences, and the dramatic consequences of mutations in such coding sequences can easily be envisaged, because if there's such a microsatellite instability occurring in a coding sequence, usually one base pair is deleted, and this immediately leads to a shift of the translational machinery in that sequence, and the protein synthesis is shifted into another gear, usually leading to the synthesis of foreign peptides after that particular mutation that usually runs rapidly into a stop codon. But still, in most instances, there is a longer or shorter stretch of peptides generated by that mechanism that hasn't been seen by the immune system before these cancer clones emerge. So clearly this leads to a loss of function in many cases of the respective genes, but in this context of this meeting, more importantly, it clearly leads to the generation of potentially cancer-specific peptides, because those antigenes are usually not generated by normal cells that have an active DNA mismatch repair system. So having that mechanism analyzed and thinking about it, we, many years back, thought that by analyzing all potentially repetitive coding microsatellites, we should be able to delineate a group of mutated sequences that are substantially supporting the outgrowth of tumor cells, and on the other hand, sequences that would be found less frequently in tumor cells would be an indicator of negative effects of the mutations, because potentially those tumor cell clones that are emerging and that have less frequently than expected mutations in the respective genes would be counter-selected. On the other hand, the ones that should have positive effects would be the ones that should be mutated for the advantage of the outgrowth of the respective cancer cell clones. And no wonder, what we found was that among the genes that had these positive effects, so that were at a higher frequency mutated, if you looked in a larger group of colorectal cancers, belonged to very well-known and established tumor suppressor genes, for example, the TGF-beta receptor type 2, or the ACV receptor type 2, or genes like BAX and many others that obviously need to be inactivated in favor of the outgrowth of the respective clones. So these genes not simply were inactivated by the mutation, but as said, also gave rise to potentially immunogenic peptides that we then further characterized in the next steps. And we asked the question whether these potential immunogenic peptides are indeed triggering an immune response in the individual patients. So we didn't have any biochemical reagents to detect that without reverse immunology. We only had these in silico data and the frequency of the mutations in the respective genes. We analyzed the expression of the respective mRNAs in a larger cohort of those tumors. And finally then asked the question, are patients who develop such kind of tumors in which these messenger RNA, or these mutant forms of the respective genes are expressed, do those patients develop a spontaneous immune response against these theoretically or bioinformatically in silico predicted antigens that I just introduced to you? So the answer to that question was that we looked first in tumor-infiltrating lymphocytes of colorectal cancer patients who were MSI positive, and indeed we found a fairly strong activity measured by ELISpot analysis against these predicted peptides that are listed here. So clearly, among the dense immunoinfiltrate that I showed you in the beginning, there are obviously T cells that recognize these antigens and that could potentially target then the respective tumor cells. We also did this in peripheral blood samples and identified among a cohort of patients who had microsatellite unstable colorectal cancers a huge reactivity in the peripheral blood against these respective peptides. And we didn't see these reactivities in patients who developed microsatellite stable colorectal cancers that were induced by chromosomal instability. And most interestingly, there's one very interesting inherited syndrome, the HMPCC syndrome. You will hear more about that by Matthias Klor tomorrow morning. And in these patients, we were wondering whether mutations carriers who never developed a cancer, although they were gene carriers, in their life, whether those patients or those risky gene carriers also were showing these immune profiles, and indeed this was the case. So I have to say that the penetrance of the HMPCC syndrome to develop an invasive carcinoma is in the range of about 50%, and we actually don't know why the other 50% that never will develop a tumor, although they have these genes, are related to tumor biological aspects or maybe even to these immunology-related aspects, which would be quite feasible. Therefore, we ask the question whether can amplification of frameshift reactive T cells actually provide protection. Of course, we can't answer this, but we put forward the following hypothesis. MSI induces the generation of the immunogenic frameshift peptides, and if a sufficient number of reactive T cells is generated early enough before an MSI lesion occurs, or is still in a stage where only few cells have developed this macrosatellite instability phenotype, potentially these activated T cells might be capable to defeat them before the emergence of an invasive carcinoma. In that regard were a couple of observations quite interesting. The following is that we had families, and this is just one, where we diagnosed in, for example, a young lady, she was a physician by herself, an endometrial carcinoma. This is kind of a hallmark for HMPCC syndrome because endometrial carcinomas never occur that early except in frame of an inherited syndrome, usually HMPCC. Now, the mother of that patient was healthy, the father was healthy, but she reported that the grandfather, the father of her mother, developed a bladder cancer at the age of 53, and there were a couple of other cancers in that family, so this smelled for HMPCC, and we did a germline analysis in that family and identified an MLH1 mutation that was the cause of that syndrome in that particular family. So look at the mother. She had a very active immune response and belonged to the mutation carriers that had displayed this high reactivity against the T cells, as I just showed to you. So obviously there is something going on. Patients who have not yet developed a tumor were obviously confronted to those mutant gene products. And then if that's the case, we have to postulate that without having a visible tumor, those patients must have generated cell clones that already have undergone this type of genomic instability, and that some kind of lesions that led to an auto-vaccination of these individuals might have been present somewhere earlier before we did this analysis. And Matthias Klor, together with Heinrich Blecker, started to analyze then a series of colorectal cancer biopsies, and what they found was that among these macrosatellite unstable cancers in Lynch syndrome patients, you find a high number of normal-looking crypts that already lost the DNA mismatch repair capacity, as for example shown here. There's a complete lack of the DNA mismatch repair activity, and these crypts already showed the profiles of macrosatellite instability, suggesting that there are normal-looking cells, normal-looking colonic crypts that already are in these mutagenic pathways, obviously or potentially producing the respective peptides and leading or potentially leading to these kinds of auto-vaccination models. So they calculated the number of crypts. This was a huge amount of mutant crypts if you calculate it over the complete gastrointestinal system. And the vast majority of these lesions have to regress because we know very well that this is not a polyposis type of syndrome, but only very few of these apparently high number of crypts find their way to go through all the progression pathway into an invasive carcinoma. So the vast majority of these initial lesions presumably are actively attacked by the immune system and presumably regress early enough. And only those that are not early enough attacked by the immune system finally find their way into an invasive carcinoma. Now, this led us to the following idea. If that's the case, then we need to identify peptides that are generated by this pathway that are shared among the vast majority of tumors that occur through this pathway. And to make a long story short, we just identified 3 peptides, which we then included into a first clinical trial, into a vaccine that was tested in a phase 1/2a trial by a subcutaneous injection into patients who had microsatellite unstable cancers according to the protocol shown here. Details of this protocol can be found on the respective website or websites that we set up for patients, where you find all further information to the details of that trial. Now, what we observed was that at least in the phase 1 trial, we, in all cases, where we could observe it because we could observe the patients for a sufficient period of time, that there was a strong T-cell response as well as a strong humoral response to the vaccine. This is just one example of a patient before vaccination, after vaccination, you can control, and here upon the vaccination, you can clearly see the increase in the ELISpot reactivity for these 3 vaccine peptides, suggesting that the vaccine is actually inducing an immune response in those patients. Whether this is will have a therapeutic effect is, of course, by far too early to tell, but at least it's an hopeful approach that we want to extend now in a phase 2b clinical trial, and if possible into a primary intervention trial where we want to vaccinate then gene carriers with the lymph syndrome. Now, to conclude, microsatellite instability is an abundant but highly inefficient process to cause cancer. The vast majority of initial lesions are regressing, as we have seen. It creates numerous potentially immunogenic cancer-specific antigenes. There are obviously shared antigenes among them. The immune surveillance seems to be an important component of the natural defense against these lesions. Only 50% of the gene carriers finally develop in invasive carcinoma. And MSI-positive cancers are a clinical example of in vivo cancer immunoediting, the concept of many people in the US, particularly Bob Schreiber, who did this for a mouse model system. So the frameshift peptides are good candidates for the development of therapeutic and preventive vaccines, and this is what we have now to test. Before I stop, I want to thank all the people who have been very much involved in these studies, particularly Elke Jäger in Frankfurt, where we did the initial clinical trial with her collaborators, and Matthias Klor, who masterminded most of this project in my lab, and many other people. Thanks very much for your attention.
Thank you, Magnus. I see already several questions. Let's start with Ugo and then Peter. Oh, the other way around.
Okay, excellent study. Did you look for the presentation of the epitopes on the tumor cells and recognition of the epitope by T cells?
We did for some of these T cells in vitro experiments, of course, where we stimulated T cells with respective peptides, the predicted peptides, generated reactive peptides, and tested that in for lytic activity, of course. Yeah, this was published in the Schwitale paper.
Now, in the tumors, Magnus, which have this defect, when you subclone the tumor, is there a heterogeneity, or does every cell of the tumor have the defect? So, of course, that would then address the question, do you have escape variants?
Yeah. So to your question, the mechanism of DNA mismatch repair deficiency is in all those cancer cells maintained. They are all microsatellite unstable. The pattern of peptides generated in the individual tumor cells is varying. But the combination of one of those 3 we identified at least in every tumor. What we haven't looked at yet is whether they are indeed expressed in all individual tumor cell clones. Yeah? But we can easily expand that pattern of vaccine peptides to a broader range, let's say 10 or 20 of those peptides. And if we're going to do this, we clearly would extend the number of clones we would be able to attack with the first hit. There's another immune escape mechanism, a very important one that I didn't mention, because the beta-2 microglobulin gene by itself has 2 repeats that are mutated in many of those tumors, because you may ask the question, if you have such a strong immune response, why does anybody get a cancer? So there must be an immune evasion mechanism involved in the pathogenicity of those tumors, otherwise those tumors wouldn't exist. And one of many answers to that question is that the beta-2 microglobulin gene is mutated during the tumor progression, usually in the invasive carcinomas, and this leads to a complete lack of class 1 expression. Interestingly, a complete one of class 1 expression hinders the tumors to metastasize through a hematogenic pathway. So there are virtually no hematogenic metastases once the tumor cells lost the class 1 expression.
Can I ask you something? Here. So the Lynch syndrome can be or can result for inactivation of different mismatch genes. And as you know, MSH6, for example, is one of them, MSH2 or MLH1. They have different signatures. And let's say MSH6 is going to produce more base pairs, whereas MSH2 will have more insertions or deletions. So at the end, the signature that we produce will result in different peptides. So did you consider that at the moment when you were designing these vaccines? No, or are you more thinking just in one type of, let's say, just singles, insertions, or just deletions, and that's it?
So the vast majority of patients who develop cancer, colorectal cancer, in frame of the Lynch syndrome, have either MLH1 mutations or MSH2 mutations. And those were included into the study. We didn't take care of the MSH6 also because they are substantially less frequently found in those tumors. Would be a different, interesting approach, yes.
Well, I don't see any other questions, so thanks again, Magnus.